Hello everyone, my name is Yifan Li and today I'm going to present my project on consensus scoring, combination of glide and ICM docking scores using data fusion. My project is conducted under the supervision of Professor Matthew Shapira at the University of Toronto. First, I'm going to start with the background and rationale of my project. Traditional drug discovery is a time-consuming and costly process, typically spanning 12 to 15 years and requiring between 1 to 2 billion US dollars. This process comprises of basic research, drug discovery, preclinical development, and clinical development. In the initial stage, basic research focuses on identifying a druggable target that can interact with the drug molecule to produce measurable biological response. Following this, the drug discovery phase involves screening a library of compounds to identify those with desired activity, called hit compounds. Once hit compounds are identified, they undergo further testing during this phase and then proceed to the next stages. Significant opportunities for time and cost saving exist within the drug discovery stage. Recent scientific and technological advances, including enhanced computing capacities and the availability of co-crystallized structures of ligands and their targets, have shifted drug discovery towards computational approaches. My project is focused on the hit identification in the drug discovery stage. There are two primary computational methods for hit discovery: ligand-based virtual screening and structure-based virtual screening. The rationale of ligand-based virtual screening is that ligands resembling an active compound are more likely to be active compared to random ligands. In contrast, structure-based virtual screening leverages the 3D structure of the target protein to predict the binding affinity of ligands and their targets. My project is specifically focused on structure-based virtual screening. The figure shows the process of structure-based virtual screening. Initially, the compound library and the receptor protein are prepared. This involves determining the structure of the receptor protein, identifying the ligand binding site, and compiling the compound library. Each compound is then virtually docked into the ligand binding site using docking software. The goal of this docking process is to predict the structure of the ligand-protein complex by exploring how the ligands can fit within the binding site. Following docking, the software assesses the binding affinity of each ligand pose and generates a docking score. Compounds are ranked based on these scores, and the compounds likely to exhibit biological activity are identified and proceed to further analysis. As previously discussed, docking software plays a crucial role in structure-based virtual screening, where it docks chemical compounds into ligand binding site of the target protein structure and generates docking scores as output. Different software use different docking methods and scoring functions. Docking methods stimulates the potential binding modes, while scoring functions use mathematical functions to estimate the binding affinity between the ligand and the target protein based on these modes. There are three main types of scoring functions: force field-based, empirical, and knowledge-based scoring function. Force field-based scoring functions are based on physical atomic interactions, while empirical scoring functions are based on a set of weighted energy terms. And knowledge-based scoring functions use statistical mechanics to predict binding affinities. Despite the development of numerous scoring functions, none achieves perfect accuracy or broad applicability. Each function has its own advantages and limitations. To leverage the advantages and balance out the errors from different scoring functions, the consensus scoring technique has been introduced to improve the probability in finding true active ligands by combining scores from multiple scoring functions. While the consensus scoring technique has many advantages compared to individual docking programs, the primary challenge is selecting an appropriate method to combine individual scores effectively, so that the true active compounds can be discriminated from others. Similar challenge is also faced in information retrieval, where researchers need to synthesize results from multiple search algorithms. In that field, data fusion metrics have been developed to combine search results effectively. It is possible that these metrics could also be applied to consensus scoring in structure-based virtual screening. In this project, I explored the application of data fusion metrics from information retrieval to consensus scoring, alongside well-established consensus docking metrics. The aim of this project is to compare the consensus docking results with results docked by individual software. We hypothesized that for any protein target, the consensus score generated by data fusion metrics will better indicate the activity of a given compound than the individual docking scores. Next, I will introduce the methods used in this study. To support our objectives, we will use two well-established docking programs that employ structure-based docking approaches, Glide by Schrodinger and ICM by MoSoft. These tools use distinct docking methods and scoring functions. Glide operates with a funnel-like strategy. It begins by generating a broad set of potential docking poses, which are then refined using Monte Carlo sampling. Glide scores these poses using Glide Score, a function that integrates both force field and empirical scoring methods. In contrast, ICM's docking approach starts with the random selection of a ligand conformation within the internal coordinate space. It then introduces a random move to the ligand to generate a new binding pose. The new pose is compared with the original pose and the optimal one is selected. These steps are repeated before a specified number of iterations are achieved. ICM scores the final poses by summation of the ICM score and the RTCNN score. ICM score is an empirical scoring function, while the RTCNN score is an AI-based scoring function. Thus, we see that Glide and ICM use different docking methods and scoring functions, allowing us to use these programs within our consensus docking strategy. To validate our docking process, we start by selecting a diverse set of target proteins. For this study, we have chosen ricin A chain, renin, cyclooxygenase 2, angiotensin-converting enzyme, and albumin kinase 1, and they should represent a wide range of enzymes. Using different types of targets in our consensus docking study can give us a chance to examine the general applicability of our consensus docking metrics. To validate our docking process, we redocked the co-crystallized ligands of the 5 protein targets using both Glide and ICM within their established binding sites. Redocking involves predicting the binding pose of the co-crystallized ligand to confirm the accuracy of our docking protocol. This step aims to de-risk our internal workflow and demonstrate the general viability of both Glide and ICM. Following the validation of the docking protocol, we docked datasets of actives and decoys for each target using both Glide and ICM. Docking actives and decoys is to use docking software to dock ligands with known activity to see if the docking software can correctly distinguish actives from decoys. Redocking and docking actives and decoys are adhering to the following protocols. For Glide, we start by importing the PDB structure. Protein preparation is then conducted using the protein preparation wizard. After that, we generate the receptor grid with the receptor grid generation package, defining the binding site as the co-crystallized ligand's binding pocket. The ligand is prepared using the leak prep package. We proceed to conformer generation using the confgen package. Finally, we submit the docking job with the generated conformers and receptor grid. For ICM, we start by importing the PDB structure. The structure is then converted to an ICM object. After that, we set up the receptor and define the binding site as the co-crystallized ligand's binding pocket. The ligand is then extracted and saved in file format. Finally, we submit the docking job. Once we obtain the docking results from Glide and ICM, which are ranked lists of compounds based on their docking scores, we will combine these scores using data fusion metrics. Before applying these techniques, it's essential to normalize the scores from Glide and ICM as they have different ranges. In this study, we will employ 4 normalization methods: the rank method, logistic rank method, Z-score method, and 0-1 linear method. The rank method involves using the rank of each compound as its consensus score. The logistic rank method, originally developed by Kauf and Salve for information retrieval, utilized a function that integrates both logarithm and logistic functions. This method assigns higher weights to top-rank compounds, which is advantageous for early recognition in drug discovery. The Z-score method and the 0-1 linear method are also common in information retrieval and have been adapted for consensus scoring. The Z-score measures how many standard deviations a data point is from the mean, while the 0-1 linear method scales the raw scores to a range between 0 and 1. After we get the normalized score, we are going to use the following data fusion metrics to combine the GLIDE and ICM score to produce the consensus scores. In this study, we are giving equal weight to GLIDE and ICM score. The data fusion metrics we used are Euclidean norm, geometric mean, harmonic mean, maximum, minimum, average, and reciprocal rank. Their formula are shown in the table. Next, we'll evaluate the accuracy of our GLIDE and ICM results, as well as our consensus scoring results using performance metrics. We employ two types of metrics: global performance metrics and early enrichment metrics. The receiver operating characteristic curve serves as a global performance metric. This curve plots the true positive rate against the false positive rate and provides an AUC value. The AUC measures the overall performance of a binary classifier. An AUC of 0.5 indicates that the model's performance is equivalent to random selection. An AUC greater than 0.5 indicates that the model performs better than random selection, while an AUC less than 0.5 indicates performance worse than random selection. Bedrock and the enrichment factor are metrics that focus on early enrichment. Bedrock incorporates a Boltzmann weighting factor to prioritize top-rank compounds in the rank list. In this study, we set the parameter alpha to 160.944, assigning a weight of 80% to the top 1% of compounds. A higher bedrock value indicates better performance. The enrichment factor measures the proportion of active compounds within a specific range compared to the expected proportion across the entire dataset. An enrichment factor greater than 1 indicates better than random selection, while a value equal to 1 indicates random selection. Then I will present and discuss the results. First, let us review the results of validation of the docking protocol. An RMSD value less than 2 angstrom indicates that the protocol can accurately identify the experimental 3D conformation of the co-crystallized ligand. Conversely, an RMSD value greater than 2 angstrom indicates that the protocol fails to predict the experimental 3D conformation accurately. Figure 1 illustrates that the docking protocol performed effectively for RTA, renin, COX-2, and ABL1 kinase, with consistent results across all 6 trials. This consistency highlights the protocol's broad applicability across different enzymes. Consequently, we proceeded with renin, COX-2, and ABL1 to the next phase, which involves docking with actives and decoys. In contrast, we did not proceed with ACE and RTA. The docking programs did not accurately predict the 3D conformation of ACE co-crystallized ligand, and we lacked an available dataset of actives and decoys for RTA. Figure 2 illustrates examples of both accurate and inaccurate predictions. In Figure 2a, the predicted binding pose aligns well with the actual binding pose, demonstrating a successful prediction. In contrast, Figure 2b shows a poor prediction, where the predicted binding pose does not match the actual pose due to incorrect function groups positioning. After validating our docking protocol, we proceeded to evaluate the performance of GLIDE and ICM as well as our consensus scoring metrics. This evaluation focuses on how well these tools can distinguish between actives and decoys by analyzing their ROC AUC bedrock, and enrichment factors. Firstly, let us examine the ABL1 kinase domain. Figure 3a presents the ROC curve for the top 1000 ligands docked with ICM, while Figure 3b shows the ROC curve for the same set of ligands docked with glide. These graphs demonstrate that both glide and ICM have moderate accuracy in discriminating actives from decoys for ABL1 kinase, and there's room for improvement. Furthermore, the bedrock and enrichment factor values suggest that glide's docking results exhibit poor enrichment among the top-ranked compounds. To enhance the overall performance and early enrichment of our docking results, we will apply the 23 data fusion matrix to our results. Next, we will evaluate the consensus scoring results using performance metrics, ROC AUC, bedrock, and the enrichment factor. First, let us examine the results evaluated by ROC AUC. As shown in Figure 4, among the consensus scoring methods using rank normalization and logistic rank normalization, only the logistic rank Euclidean norm demonstrates a superior performance compared to both glide and ICM, increasing the ROC AUC value to 0.74. Figure 5 shows the ROC curves of consensus scoring metrics that use Z-score normalization and 0-1 linear method normalization. We can see that only Z-score harmonic mean performed better than both glide and ICM, increasing the ROC AUC value to 0.74. In summary, both the logistic rank Euclidean norm and the Z-score harmonic mean improved the ROC AUC value of our individual docking results. In addition to overall performance, several metrics also improved early enrichment. Figure 7 presents the bedrock values for glide, ICM, and consensus scoring metrics with improved bedrock value. Notably, Z-score Euclidean norm and maximum significantly increased the bedrock value to 0.49, while Z-score average and 0-1 linear method maximum increased the bedrock value to 0.41. Figure 8 shows the enrichment factors for glide, ICM, and metrics with improved values. Note that values marked with an asterisk represent the maximum possible enrichment factor for the dataset. The results show that both the Z-score maximum and 0-1 linear method maximum achieved notable improvements in enrichment factors. Given that we have three different performance metrics, determining the best consensus scoring metric can be challenging. A consensus scoring metric may perform well according to one performance metric but poorly according to another. To address this issue, we will utilize a 3D bubble plot. This approach allows us to visualize and consider all three performance metrics simultaneously, providing a more comprehensive evaluation. Figure 9 presents the 3D bubble plot for the ABL1 kinase. In this plot, all matrices except the 0-1 linear method Euclidean norm demonstrate improvements in docking results. On the plot, moving up the y-axis indicates higher ROC AUC values, while moving right along the x-axis represents higher bedrock values. The size of the bubble reflects the enrichment factor, with larger bubbles indicating better performance. From this visualization, we can determine that the optimal metric is located in the upper right corner of the graph and has a large bubble. For the ABL1 kinase dataset, the Z-score maximum is closest to the optimal position with large bubble, suggesting it performs the best. It is important to note that the logistic rank Euclidean norm and Z-score Euclidean norm also performed fairly well. We can also apply the 3D bubble plot to COX-2 to identify the most effective metric. Figure 10 reveals that the Z-score Euclidean norm performs the best for COX-2. Additionally, the Z-score maximum, Z-score geometric mean, Z-score average, and glide also demonstrate strong performance. For renin, the Z-score Euclidean norm performed the best. Additionally, the 0-1 linear method harmonic mean, 0-1 linear method maximum, and z-score maximum also demonstrate strong performance. Based on our 3D bubble plot analysis, we determined that the z-score Euclidean norm and z-score maximum performed the best among the 23 proposed data fusion metrics. Figure 12 shows the bedrock values for GLIDE, ICM, and these two consensus scoring methods. Our analysis confirms that both the z-score Euclidean norm and z-score maximum significantly enhanced early enrichment across all three targets. Figure 13 demonstrates that applying the z-score Euclidean norm and z-score maximum also improves the 0.1% enrichment factor value. Note that values marked with an asterisk represent the maximum possible enrichment factor for the dataset. The improvements of early enrichment by these two data fusion metrics make them valuable tools for future virtual screening. Finally, I will conclude the presentation by discussing the limitations and outlining potential future directions. The first limitation of this study is related to the choice of docking programs and targets. Both GLIDE and ICM use empirical scoring functions, which may introduce errors and biases into the consensus scores due to their similar scoring mechanisms. Additionally, since all three targets in this study are enzymes, the applicability of our docking protocol and consensus scoring strategies to other protein types, such as membrane proteins, remains uncertain. The second limitation is the equal weighting of GLIDE and ICM docking scores. Depending on the target protein type, it may be beneficial to assign higher weight to the program that performs better of that specific target. To address these limitations, future research will explore other docking programs and targets to refine consensus scoring methods. By identifying which programs are more effective for a particular type of targets, we can adjust the weightings accordingly. Furthermore, with the rapid development of artificial intelligence, there is potential for AI to optimize consensus scoring strategies by selecting the most effective docking programs and scoring methods based on the property of the target protein. Finally, I would like to express my heartfelt gratitude to my supervisor, Professor Matthew Shapiro, and Dr. Conrad Verenzo for their invaluable guidance, support, and encouragement. My sincere thanks also goes to the Structural Genomics Consortium and the Department of the Pharmacology and Toxicology at the University of Toronto for giving me the platform to conduct this research project. I would also like to thank the graduate students Angela, Lakshi, and Avian for their willingness to answer my questions with patience. Thank you so much for listening, and here are the references. This is the end of my presentation.